Good afternoon. All right, so we have been, I know a lot of announcements, I guess, today, but we last class, we're working our way through chapter 7, finished that up. So we finished replication, transcription, and translation. I'll say students tend to get the processes of those three things kind of mixed up in their minds. So you want to make sure that you kind of have those three things separate, can describe, you know, the beginning, the middle, and end of all of them. Your homework will help you with that. So you can be working on chapter 7. I would encourage you to do that. Not very many of you have. So you might think about starting that pretty soon this weekend if you haven't already. We finished last time talking about ways that the DNA can be altered in some way, the effects of mutation, and the 3 kinds of horizontal gene transfer. And then we began our time thinking about viruses, some basic characteristics, morphologies of viruses. I want to just mention that the shape of a virus doesn't have anything to do with what kind of disease that it causes. So we had the 3 different shapes of the capsids, helical, icosahedral, or complex, and then kind of added in whether the virus had an envelope around it as well. So thinking a little bit more closely about that viral envelope. And I will say that it's very common for viruses that infect animals to be enveloped. So most, and when I talk about animal viruses, we're animals, right? So most animal viruses are enveloped. There's lots that aren't, but I would say the majority are. Okay. And there is one bacteriophage. So bacteriophage are viruses that infect bacteria that is enveloped, but most bacteriophage are not enveloped. Where do they come from? Remember, viruses tend to steal lots of things from their host. That envelope is always going to be acquired from the host. So when you think about the composition of the viral envelope, what are our membranes composed of? It's a phospholipid bilayer. So that's always going to be the composition of those membranes. They might have some glycoprotein spikes on the surface. So for instance, the influenza virus has a couple of different kinds of spikes on the surface, and we're going to talk more about those later in the course when we talk more specifically about influenza virus. Non-enveloped virions can have spikes on them as well. Those spikes are important in recognizing the kind of host cell that it needs. So there's very specific receptors there. So important in attachment, in entry. And some of the spikes on influenza virus are important when the virion leaves the cell so that it's detach in the right way. So if sometimes these glycoprotein spikes, sometimes you'll see those referred to as peplomeres. I'll always just call them viral spikes. We've heard that term a lot lately because, of course, coronavirus has spikes. And typically that's, if a virion has spikes, that's the part of the virion that our immune system is best at recognizing. So, I'm pretty sure, there's a couple hundred different coronavirus vaccines out there in various stages. I think almost all of them incorporate some kind of, viral spike protein in some way. And if a cell doesn't have spikes, or sorry, not a cell, if a virion doesn't have spikes, not all of them do, those functions, recognizing host cells and attachment and all that will be embedded into the capsid somehow. So they'll have, you know, receptor proteins in there. Okay. One other thing that I wanted to bring up here, in terms of what kind of virions are hardest to kill, enveloped virions are generally going to degrade very quickly in the environment. So thinking about, well, how long can a virus stay on a surface and still be able to infect us? That's a really important question. And in general, if a virion has an envelope, you might think that that would protect it, but it's a phospholipid membrane. It dries out really quickly. Okay, so that's going to degrade really quickly, and most envelope virions are going to only last in terms of maybe a few hours to maybe a day, maybe 2 days at the most. You know, with all sorts of things, there's exceptions to it, but generally speaking, envelope viruses are pretty easy to kill. So last summer, I read an article that said coronavirus stays viable up to 25 days on surfaces. And I thought, you know what, that just doesn't sound right. It's coronavirus has an envelope, and you know, you want to read things skeptically. If something doesn't sound right, see if you can find out from another source. You know, where's the source coming from? We now think that, you know, you don't need to store your groceries in the garage for 5 days or whatever until you use them. The coronavirus just doesn't stay viable for very long on surfaces. So that's good. Now, naked viruses, right? They're a lot tougher. They've got a protein shell that just withstands, you know, variations in the environment, drying out a little bit better. And anecdotal evidence, one naked virion is the Norovirus. So I don't know, probably a lot of you have had Norovirus at some point in your life because of a really nasty enterocolitis. So throwing up, diarrhea, maybe both at the same time. It's really awful. And that hit our family a number of years ago. My husband and I were both, you know, had it at this exact same time. Eventually we couldn't see straight. We just moved here. We didn't know anybody. So we ended up calling the emergency squad, and the paramedics came in, and our kids who were in their cribs had been thrown up, and we didn't even know it. That's how sick we were. So it was just awful. And then a week later, my parents came to visit from out of state, and they got the same thing. So that stinking norovirus, no envelope on there, was viable on some door handle or something. And, you know, that's just, again, anecdotal, but that's the way that I always remember that. Okay, so we were talking about those viral spikes. There's always going to be something on the outside of a virion that's going to recognize the appropriate type of host cell for that virion. And oftentimes viruses are going to be very host specific. Pretty much any living creature has viruses that are specific for it. So for example, tobacco mosaic virus, it's specific for tobacco plants. You know, we're not going to get ill with tobacco mosaic viruses. Those receptors just don't match up. There are lots of viruses that are specific for bacteria, for a specific species of bacteria even, okay? And here's an example of a virus that infects humans, HIV. Of course, there's HIV in chimps and some other animals. HIV is specific for certain kinds of cells in humans. So you may have heard the helper T cells, the CD4 positive T cells. These pink dots are the HIV, and the huge gray background, that's a T helper cell. HIV can infect some other types of cells in our body, but it doesn't do those cells any damage, mostly immune cells. If we swallow HIV or breathe it in, it doesn't do us any damage at all. So kind of specific ways that we have to be infected with that. There are viruses that also infect protozoa. There are viruses that infect fungi, but we think that fungi can only pass it to each other when their hyphal tips fuse. Fungi have a really tough cell wall, and apparently viruses don't penetrate very well through that. There are some viruses that are considered generalists, meaning they can infect lots of different species. West Nile virus is one, birds, humans. It's quite an assortment of different animals that can get West Nile. I would say coronavirus would probably be considered a generalist. We found tigers with COVID-19 and pets, apparently kind of rarely, but okay. So still thinking about characteristics of viruses here. We talked about all viruses have either DNA or RNA, right? They don't have both, but they have some kind of nucleic acid. Okay, they can have double-stranded or single-stranded DNA. So of course, single-stranded DNA, that's a form of nucleic acid that we don't find in any life. So that was kind of mind-blowing when we discovered that. There are RNA viruses that are double-stranded, which again, that's kind of crazy. We don't find that normally, or single-stranded RNA. That nucleic acid can be circular or linear. So in this graphic over here, there you see the red lines, that's the linear, whatever it is, DNA or RNA. If it's linear, that can be considered either segmented or non-segmented. I think sometimes that's also referred to as continuous. So what's the difference there? Well, in this picture here, this is segmented nucleic acid, whatever it is, because you've got 3 different segments there. If it's continuous, you would have your DNA or RNA all in one single strand. Okay. What's the difference? RNA viruses tend to mutate really quickly because the polymerases that they use just aren't they don't have error fixing mechanisms, and they're just more indiscriminate about throwing the different base pairs in there. So say an RNA virus makes a mistake when it's copying its RNA right at the beginning, and it throws off, say, the reading frame, and all the rest of it, is not right, then that can be a problem. But if the RNA is in segments, part of one segment doesn't copy correctly or transcribe properly or whatever, then maybe the rest of it is going to be okay, and the virion might still be able to infect. So just some differences there. The gene number is small. So how small is it? Well, when we sequence the human genome, before we finished that human genome project, we thought humans had at least 100,000 genes, maybe more. After we sequenced it, we found, just from the sequence of bases, you can't tell for sure what's a gene and what's not, but we've now revised that to be a much lower number, somewhere between 25,000 and 40,000 genes. Can't be precisely sure at this point. Compare that to a bacteria like E. coli, somewhere in the neighborhood of 4,000 genes, sometimes a little bit more than that, depending on what strain we have. Compare that to a virus. The smallest genome of any virus is the hepatitis B virus, and it only has 4 genes. And that ranges from 4, I think herpes virus is one of the bigger ones. It has between 100 and 200 genes. So still at least a magnitude, maybe 2 magnitudes smaller than the bacteria. But that makes sense because remember, viruses enter into other living cells and just use everything that that living cell has. And so, little story, I sometimes, one of my nicknames for my son is hepatitis B virus. And that sounds kind of weird, but he tends to pack light. So he came home from fall break a couple of years ago, and he got a ride back to Grand Rapids, and we went and picked him up at his friend's house, and he had his laptop and a football with him. And he was coming home for 4 days. And I said, oh, where's the rest of your stuff? And he said, oh no, this is all I brought. You know, I did that little mom sigh. But when I thought about it, I thought, oh, this is just like a hepatitis B virus. Because he's coming home, he's got a bed, he's got a towel and shower, he's got food, he's got old clothes, he's got an old toothbrush. Everything he needs is at home already. Okay, just like the hepatitis B virus, it's going to enter into cell and just use all the cell's machinery. It really only needs a couple of things for it to replicate itself. So I don't know if he appreciates that very much when I call him that, but anyway. So this I don't think I posted this, but this is just a picture from your book. We can break open cells and treat them so that the DNA is visible. And you can see here the viral genome. There's a virus that's infected E. coli. The viral genome is just really tiny, and all the rest of the stringy stuff is just some the partial genome of E. coli. So, okay, much smaller genomes. All right, what sort of functions do viruses need to bring with them? Well, things that the cell wouldn't normally do. So maybe there needs to be some instructions for making viral spikes, for instance. That makes sense. There might be some regulation of the host cell that's necessary, turning some genes off, some genes on. That's reasonable. And they may need some proteins, some enzymes to replicate their own genetic material that we as hosts wouldn't have. So I put this little reminder down here because sometimes I talk about viruses before we talk about genetics. So remember, this is the flow of information in all of life, basically. And if it's an RNA virus, it's going to need to make RNA from RNA, right? And that's something that we never do, or that bacteria never do. All right, so that's an enzyme called replicase. Some viruses, of course, start out with RNA and make DNA from RNA. That's something that we never do. So if that's a virus's strategy, it has to bring in its own enzyme to do that. And that's called reverse transcriptase. Okay, that was another mind-blowing thing when that was discovered. Late 70s, early 80s. All right, so taking all that information, kind of that basic information that we know about viruses, we can now think about how are they classified. There's really two different schemes that we use for classifying viruses. One we call the hierarchical, difficult word to say. And the other scheme is called the Baltimore Classification. They're both used. The Baltimore Classification is named for David Baltimore. He's the one that first discovered that reverse transcriptase. He's done a lot of work with HIV. So this scheme is a little bit newer, and it's a little bit less subjective. So in this scheme, we classify viruses just based on their nucleic acid. So do they have double-stranded DNA or single-stranded DNA or whatever? Don't memorize which is which, but you know, it assigns a class based on what the nucleic acid is like. And then these down here at the bottom, single-stranded RNA retroviruses, they're the ones that have that reverse transcriptase. And then there's just a few viruses that don't replicate their DNA in quite the same way. They go through an RNA intermediate to replicate their DNA. So kind of similar thing there. With the hierarchical classification, we are trying to treat viruses like they're living organisms. So we'll talk about that a little bit later. Are viruses alive? There's some controversy about that, but we'll talk about what it means to be alive and see if viruses fit in there. But this is a little bit more subjective because, of course, viruses don't really meet some of the criteria for being alive, but we try to put them into the same classification. So there's no kingdoms or phylums or classes for viruses, but all of them are put into one of 3 different orders. And then below that, all viruses belong into a family. And we know which family they're in, or we know by looking at it that it's a family name because it will end in viridae. Okay, and it's correct grammatically to italicize family, genus, and species of viruses, just like it is for other living organisms. Okay, so for example, Herpes viridae, okay, that's the family that the herpes virus is in. The genera then carry the suffix virus. So herpes virus is in the family Herpes viridae. Species, I didn't write this down here because it's sometimes a little bit hard to pin down, but usually a viral species is just named for the disease that it causes. So I gave an example down here, one that's not known to me, but here's a family of virus, Bunyaviridae, a genus Phlebivirus, and the disease that we get from that Phlebivirus is Rift Valley fever virus. Right, so if you're talking about it as a species, you capitalize it and italicize it. If you're just talking about the disease, then you don't need to italicize it. So there's a lot of little rules there. If you go to the CDC website on the grammar of emerging diseases, it's like 20 pages long. So it gets really complicated. Sometimes it's easier to remember because you kind of carry the same name through it all, like the herpes virus. So all of those classifications are based mostly on these 4 things. And again, it's a little bit subjective. You know, we learn more and we might change the classification of a virus. But you look at the nature of the nucleic acid, the symmetry of the capsid. So is it icosahedral? Is it helical? Is it considered complex? Is there an envelope? The size of the virion itself in terms of how many base pairs is the nucleic acid and the capsid. So those are just some of the things, again, the shared characteristics that we would use to put it in there. And I included this picture, again, not to memorize any specific details, or maybe I didn't include it in your packet, I forget. But just to kind of appreciate, hey, here's the diversity, here's the things we use to classify, kind of see how it all works out together a little bit. So this graphic has the nucleic acid at the top, the symmetry of the capsid, whether it has an envelope or not. So the genome architecture, is it single-stranded, double-stranded? Is it, you know, circular or segmented or whatever? It also has the Baltimore class on there. Nice little pictures there, includes the family and just some other information. So again, genome size, you know, smallest one here, the Hepadna viridae, that's a hepatitis B virus, the smallest one. Herpes is pretty big. This Iridoviridae, I'd never heard of. I looked it up, and it's a viruses that only infects like reptiles and amphibians. So anyway, that's just kind of some diversity there. All right, so let's get in probably, you know, the part that's a little bit, maybe a little bit more conceptually difficult is the replication of viruses. There's two different ways that viruses will replicate. And we'll talk about it first in viruses that infect bacteria, bacteriophage, and then we'll think about a few things that are a little bit different as far as entry and release with viruses that infect animals. Okay, usually it's going to, any kind of replication is going to result in the death of the host cell. Perhaps not right away, but eventually it would. And in terms of our first kind here, lytic replication, we're gonna describe that in 5 different steps, all right? So your picture in your book goes through this pretty nice little figure here. Okay, this is, we're using it as an example here, a T4 bacteriophage. So phage means eater, bacteria, eater of bacteria. The T4 bacteriophage, it's a double-stranded DNA bacteriophage. Most bacteriophage are double-stranded DNA. And this is one that only infects E. coli. Okay, and we consider it a virulent phage. And that just means that it only does the lytic, lytic replication. Okay. So here's a picture of T4 up here. It looks kind of like a lunar lander, kind of an odd structure. It has kind of a elongated icosahedral capsid there, along with a tail sheath and these kind of spindle fibers here. Right at the ends of these spindle fibers, there are receptors that recognize, you know, something about the outer membrane of E. coli. Okay, so you can see this is definitely a gram-negative cell wall that it's attaching to. Bacteriophage do not have any kind of purposeful motion, right? They don't have flagella. They can't move towards some attractant. They're just going to randomly, when they randomly land on an E. coli, okay, they will attach. And then, so that's the attachment phase. Entry is when we get a contraction here of that tail sheath, and that double-stranded DNA gets basically kind of injected inside of the cell. But it needs a little help. Think about it, DNA is not like some stiff thing that's just going to poke in there. They need to do something, release a chemical that's going to weaken the cell wall a little bit. I mean, there's not a lot of peptidoglycan that would be kind of rigid there, but you need to break that up a little bit. The bacteriophage carries with it a protein called lysozyme. And lysozyme weakens the peptidoglycan in the cell wall. Okay. It actually breaks the bonds between those alternating sugar units, NAG and NAM. Yeah, okay, so those glycosidic linkages. All right, so we weaken the cell wall a little bit so that that DNA can be injected into the cell. Upon entry, and then the capsid and the tail fibers and everything, they just fall off. Their work is completed. The phage DNA will start to degrade the bacterial chromosome. So you can see all the little pieces of the bacterial chromosome over here. And in the synthesis phase, that DNA has been transcribed and translated. So it's making all of the parts that it needs to assemble new viruses. It's also synthesizing some more lysozyme to be included in those new phage. Okay, and so here's all the parts that are synthesized. All those parts just get assembled spontaneously. You would think there might be some enzymes that would need to mediate that, but we think it all just kind of comes together. And then the lysozyme, once again, is important to weaken the cell wall. So this lysozyme comes into play in entry and release. Okay, so those virions then are going to lyse the cell. That's why it's called the lytic replication cycle. Make sure I didn't forget to say anything there. Okay, so I kind of like this picture down here. This wasn't in your book, but you got to kind of feel sorry for that E. coli. All right, it can be actually infected with multiple bacteriophage all at once. Obviously it's lysing, never stood a chance there. Usually somewhere between 100 to 200 bacteriophage are going to be released there. Okay, and if you look at we could say, we do growth curves with bacteria. If you look at a growth curve, assuming you have one bacteriophage that enters a cell for a long time during the synthesis and assembly, right, you're not going to see any increase, but then all at once you have a burst, right, that lysis of the bacteria cell somewhere between 100 to 200. And look at the time here. It takes about on the order of 25 minutes. Okay, so that's kind of an average bacteriophage, how long it takes to do the lytic replication. Okay, of course, if this was a bacterial growth curve, we would have a lag and more of a steady, exponential period up. Okay, so bringing in something from the media here, I always like it when they try to talk about something sciencey in the newspaper, and sometimes they get it kind of close, sometimes not really. But this was an article in the Wall Street Journal in 2016, so a little while ago. I said at the beginning of this section that last time that viruses really, what good are they? They cause disease. There is potential to use viruses that infect bacteria to control numbers of bacteria, especially during infection. So this article basically is talking about the problem of bacterial resistance to antibiotics. And of course, that's still a problem now. It's not the most important public health problem we're facing, so we haven't heard much about it lately. But this attempts to explain how, bacteriophage replicate. So, they land on a bacteria and inject their genetic material. And then I love this. It says baby phages take over the bacterium's body. Right? So, baby phages don't grow up into big phages, right? They're just assembled. We talked about that, but, all in all, it's fine. They do, then they burst out of the cell. So the whole article is about the potential of using this to control bacterial infections in burn victims. We have a really hard time controlling that, because obviously when you lose your kind of first layer of protection, you're really susceptible to bacterial infections. So they were dipping strips of gauze in bacteriophage that were specific for Pseudomonas. Pseudomonas is a really common hospital-acquired infection for burn victims. And they're having really good success controlling these infections where antibiotics weren't necessarily doing the job. So this has been a technology that's been around for more than 100 years. You can take bacteriophage in pill form, more commonly if you live in like Eastern Europe or Russia. Once we came out with antibiotics in this country, we didn't really rely on this very much, but we may find that we can go back to this. We're really kind of slow to accept this in this country because just the fact of taking viruses on purpose is kind of a scary thought, but it has potential. All right. Okay, second kind of replication is that bacteriophage do is called lysogenic replication. And in this form of replication, the cell's not killed right away because the genetic material of the bacteriophage actually incorporates into the host cell and just hangs out there for a while. Okay, so here we're using as an example lambda bacteriophage. You can see it looks a little very similar to that T4. There's no spindles there. The receptor proteins are just at the end of that stalk there. Lambda is considered a temperate phage. Okay, oops. And that just means it can also do the lysogenic cycle. Okay, so temperate phage. Sorry if that's kind of hard to read. So virulent phage do only the lytic cycle. A temperate phage can actually do both lysogeny and the lytic replication. All right, so here we get the lambda phage just, by random motion landing on, this is also specific for E. coli. Okay, and the entry, you can see the double-stranded DNA entering into the bacterial cell. And then when it goes into the lysogenic cycle, you can see that the bacterial chromosome here is not being degraded. Okay, actually the phage DNA incorporates into the host chromosome. Okay, so we call that phase the prophage. Okay, when the again viral DNA is incorporated into the host DNA, and it also will, the phage will also release some proteins that down regulates the ability of the cell to have another prophage. So basically, it makes it harder for another temperate phage to come and infect the cell at the same time. That you can see how that would be an advantage. You wouldn't want a whole bunch of viruses incorporating into that chromosome at the same time, because eventually the cell's not going to be functional anymore. Okay, so you really only want one at a time. And then every time the cell divides, that chromosome, including the bacteriophage, is going to divide at the same time. Okay, so the infected cells grow normally for generations. At some point, then the viral DNA can leave the host bacterial cell. And we call that process induction. So the viral DNA leaves the host chromosome. When does that happen? Well, we're not exactly sure, but we do know that if the cell is in danger of dying, so if it's in some kind of harsh environment, that will stimulate induction, okay? And that just means, again, the host chromosome degrades. Now we're entering into the lytic cycle. So the viral DNA comes out, synthesis of viral parts, okay, that's assembled, and then we get release. So it kind of enters back into the lytic cycle. One other thing to mention about this, I've got this term down here, lysogenic conversion. We know that it happens sometimes that when the virus incorporates into that host cell, that actually can change the phenotype of the host cell. Okay, so the phenotype of the host cell is altered. So what do I mean by phenotype? So for you and me, it's observable characteristics. For bacteria, a phenotype could be, well, can it use, this nutrient or can't it? The instances where we know where this has happened is that we know that some bacteria are enabled to make some kind of enzyme or some kind of toxin that it can only make if it's been infected with a bacteriophage. So for example, the organism that causes diphtheria, that's perfectly harmless unless that bacterium has been infected with a bacteriophage that gives it the ability to make diphtheria toxin. Okay, then it's this deadly disease. Same thing with Streptococcus pyogenes. If you're in lab next week, or you used it this week in lab, but you're going to talk about Streptococcus next week. If that organism has been infected with a bacteriophage, which happens rarely, then it's able to make enzymes that can cause the patient to have a bright red rash. Okay, that's known as scarlet fever. All right, so we know there are instances where, again, it changes the phenotype of the cell. Okay. All right, so questions. Okay, so we'll talk about, there are some differences when we're talking not about a bacteriophage, but about a virus that infects animals. It's the same basic pathways, lytic and lysogenic pathways, but there are some differences because lots of animal virus have envelopes. So for instance, how are you going to get lysis of a cell if you need to get a membrane around each one of those viruses that leaves the cell? Okay, that really can't happen the same way because it has to be able to pull its membrane from the host cell somehow. The eukaryotic nature of animal cells, that's a kind of enigmatic way to say, remember, animal cells have a nucleus. So like a DNA virus needs to go where the enzymes are to replicate its DNA. So that's going to be, has to have a way to get through that nuclear membrane as well. And then, of course, remember the animal cells lack what that bacteria have. They are going to lack a cell wall. So that's, again, going to be differences in how it's going to enter a host cell, how the virus will enter, and how it will leave again. All right, so 3 mechanisms of entry of animal viruses. So pull this figure from your book. And I've used this figure for a long time, and I just realized that there's an error in it. In the 6th edition, they fixed the error, but I didn't notice that there was an error. So it's talking about entry of animal viruses. And then right here, it talks about the phage genome. Okay, so let's assume that we'll just call this the virus genome, because again, we're thinking about animal cells here primarily. This isn't incorrect what it said, but we're kind of shifting our focus here a little bit. This is the way that phage would enter a cell. We call it direct penetration. So, you know, there are some phage that basically are just a capsid that don't have those spindle fibers or tail spindles. The little red dots here, that just means those are the receptors on the cytoplasmic membrane. Of course, the phage would be attaching to the cell wall of the bacteria, though, so I guess that is not quite correct. But an animal virus that doesn't have the cell wall, this is the way, one of the ways that it can enter. So just, you know, it kind of, the capsid fuses with the receptors, you know, a pore opens up, and the viral genome is injected inside the cell. Okay, so this is going to be naked viruses. Okay, one method that they could have to enter. And only naked viruses, a membrane-bound virus wouldn't enter a cell this way. There's membrane fusion. Okay, so you can have a virion that fuses, a membrane-bound virion that fuses its membrane with the cell membrane of a host cell. So that membrane gets left on the outside. The capsid and the virion, the capsid and the nucleic acid enter the cell. The capsid falls off pretty quickly, and then you're left with the viral genome. So this is going to be enveloped viruses only. Obviously, membrane fusion has to have membrane to fuse. And then the third method of entry that can be either one, either naked or enveloped, can enter this way. Endocytosis. So remember, animal cells can some of them can kind of extend part of their cell membrane to surround something. And lots of viruses will do this. They will trick the cell into endocytosing them. Okay, so the whole membrane, everything, or again, if it's non-membrane, this can happen as well. The membrane and the capsid are uncoated basically once it's inside of the cell, and you're left with that viral genome. Can you think of what the advantage might be if you are a membrane-bound virus? There is an advantage to endocytosis, to getting into a cell that way, rather than by membrane fusion. Okay, and the reason that that can be a little bit of an advantage, you see here, when it's just kind of swallowed up by the cell, it doesn't leave any evidence that that cell has been enveloped by a virus. So here you're seeing, viral glycoproteins are going to remain on the outside of that cytoplasmic membrane. That's a big advertisement to our immune system, our innate cells. Hey, look, this cell's got something going on that shouldn't be in there. Okay, here with endocytosis, how are cells? There's nothing on the outside that would alert our immune system that cell has been infected. And we'll learn more about that when we talk about our immune system. There are ways that that's advertised, but it kind of comes a little bit later on in the infection, not kind of our first responders. All right, so those are the three mechanisms of viral entry. A virus has to have a strategy then to make more copies of its genome. So we talked about the different permutations of nucleic acid here. If a virus is single-stranded RNA, and we touched on this a week or so ago, there's two possibilities. It can be a plus strand of RNA or a minus strand. Okay, so it makes a big difference. Remember, plus RNA, that's the same thing as messenger RNA. So that's ready to be read at the ribosome. So it functions as messenger RNA. Negative sense RNA basically has to be copied to positive sense RNA. So we'll see shortly. There's a nice graphic that kind of shows how that works. Okay. So there's lots of examples of both positive sense and negative sense RNA. That's lots of viruses. And one other thing to say here, just keep in mind that if it's a DNA virus, they have to go where we replicate our DNA in the cell. So they have to go into the nucleus. And generally, RNA viruses don't need to go into the nucleus. There is an exception or two to that rule, but generally they're just going to stay in the cytoplasm because, you know, they don't need DNA polymerase 3 for anything. Okay, so that's, again, there are exceptions to that rule. All right, so this is the graphic here that kind of walks through how a cell would copy their whatever, RNA, I guess. And it doesn't talk about how it would copy its DNA because that's going to be exactly how we would do it. But if you have a positive sense RNA virus, again, that single-stranded positive sense can be used straight at the ribosomes, can be read at the ribosomes and made into proteins itself. But of course, it needs to synthesize more copies of itself, and it has to do that through a negative sense intermediary. So we can't make, or it can't make positive sense RNA from positive sense RNA. It makes the complementary negative, and then it can make more positive from that. Okay, some examples of positive single-stranded RNA viruses. Coronavirus is a big one there. Poliovirus was another example. Negative single-stranded RNA viruses, it's not a whole lot different. They have to make the complementary copy of that positive strand, and then so then that positive sense can be used at the ribosomes, and that's copied again to make more of its genome. Let's see, is it HIV? Rabies, Ebola, flu, those are all negative sense single-stranded RNA viruses. And then there's this weird double-stranded RNA where the strands are unwound, the positive sense used at the ribosome, and the negative sense, you just kind of make a copy of that to make it double-stranded again. That's a lot more rare. I just jotted down an example of that. Reoviridae, that's not something you need to memorize, but Rotavirus is a Reoviridae. That's a pretty nasty diarrhea that very young children and babies get that can be kind of serious. I think we have a vaccine to that now, though. All right, so again, not to memorize individual viruses, but I just like this graphic because it reminds us that no matter what kind of nucleic acid the virus has, it has to have a way to get that positive sense RNA. So it has to have the right enzymes or use the host enzymes, either way, to get their nucleic acid in a form that can be read at the ribosomes so it can start making its own proteins. Okay, so just give some examples there. All right, so assembly and release. A few differences here from the bacteriophage. The timing of it usually takes a lot longer for viruses that infect animals. Remember how long the bacteriophage, we said 24 minutes, I think 25 minutes. Animal viruses, approximately 25 hours. Okay, about a day, right? So typically takes a lot longer. Not sure why exactly. And then, let's see, some different ways that release from the host cell happens. Naked viruses can be released through exocytosis. It's the opposite of endocytosis, right? Or lysis. Your book doesn't give a graphic to show that, but remember, if it's a naked virus, it doesn't need to acquire any kind of membrane, so it can just be kind of, released from the cell. And an enveloped virus can either be released by exocytosis. Let's see, spell that wrong. Or a process, it's a little bit slower generally, that's referred to as budding. So it really just depends where those envelope viruses are pulling their membrane from. If they're pulling it from the cell's cell membrane, it's typically going to bud out. If it pulled its membrane from the nuclear membrane, then it just needs to be transported out of the cell, and that would be by exocytosis. All right, so here's your graphic of budding in envelope viruses. The virion will express any kinds of proteins that it needs to be in that membrane on the surface of the cell here. And then as it grabs some of the cell membrane, it will take that with it. Okay, so this can be kind of a slow process, maybe a very persistent infection. If this happens fast enough, it can pretty much bring about the same thing as lysis, because a host cell can only stand, losing so much of its cell membrane before it's not going to be able to function anymore. So again, it can be slow or sometimes fast enough to kill the cell. So if you look at the, again, kind of a growth curve of viral numbers in a persistent infection, so that's when you have probably some kind of budding taking place. This release of virions is going to be fairly slow over time, probably eventually kill the cell here. And just comparing that to our bacteriophage, where release was all at once during the lytic cycle. All right, so viruses that infect animals, okay, also can do the lysogenic replication cycle, but for animal viruses, we don't call it lysogeny, we call it latency, okay? So that's when animal viruses remain dormant in host cells. This is just basically lysogeny in animal cells. It's called latency. Okay, so remember during that lysogenic cycle, that virus may be incorporated into the host cell for years or for a long time and not really do any change. So it may last for years with no viral activity. And sometimes in animal cells, those viruses will incorporate right into the animal cell chromosome. Sometimes they incorporate in little plasmid-like structures. So maybe incorporated into the chromosome, or as a plasmid-like structure. Okay, herpes virus is one of the viruses that does that. So there's lots of different forms a herpes virus infection can take. Of course, cold sores, genital herpes. That virus always incorporates into the host cell, actually as what we call, we're going to talk more about this later, an episome, just a little plasmid-like structure. So even though it's not into the chromosome, whenever the cell is replicated, that herpes genome gets replicated as well. Okay, so that's an infection that we say is latent. It can, flare up from time to time, and in between flare-ups, the host is not having any issues at all. One form of herpes infection is chickenpox. And of course, that can reactivate years and years after the person has had chickenpox to a very painful rash called shingles. And we'll talk more about that later on as well. Of course, chickenpox rash is kind of itchy, and annoying, but shingles is quite painful when it reactivates. Okay, and that incorporation into the host cell is, for animals, going to be permanent. Okay. Okay, so I just brought up that slide of the lysogenic cycle as well. I'll refer back to that in a second, but I just wanted to compare, just to be clear, lysogeny and bacteriophages with latency in the animal host. Of course, that we talked about how it, in lysogeny, it forms a prophage. Okay, so that was right here, the prophage, when the virus has incorporated into the bacterial cell. Of course, it wouldn't make sense to call that stage in latency a prophage. We call it a provirus. So that's, again, when the viral genome is incorporated into the animal cell. And we talked about how it can leave the lysogenic cycle and go back into the lytic cycle in a process called induction, when it leaves the lysogenic cycle. And I mentioned for animal cells that provirus is always going to be permanent. Okay, so there's no induction, unfortunately. So once we are infected with a virus that can become latent, as far as we know, there's no way to ever get rid of it. So HIV is one of the viruses that will incorporate into our chromosomes, and it's one of the reasons we haven't been able to cure HIV. Once it's there, there doesn't seem to be any way to get that genome to leave again. All right, so just some differences there. All right, so any questions? Don't be afraid to jump in if you have questions about something. Thinking about, well, how do we work with viruses in the lab? Are you likely to see any viruses in 213? No, because remember, you have to grow viruses in a living cell. At one point years ago, we did a lab, we used to do a lab where we would grow bacteriophage. We had trouble getting the results to be consistent, and so we kind of gave up on doing it. But you can culture viruses in bacteria, but you can't culture viruses just on growth medium, right? So remember, viruses have to be in a living cell in order to reproduce. But we can grow them in cells, and how that's typically done is that you would make a lawn of bacteria on some kind of growth medium, let that even confluent lawn of bacteria grow up, and then you add your solution that has the bacteriophage in it, kind of spread it over the whole plate, incubate those cells some more, and the bacteria that have been infected with the bacteriophage, you'll see kind of an empty spot where all the bacteria cells have been killed because they've been lysed. So we call those plaques. So if we want to assay how many bacteriophage we have in a sample, this is the way you could do it. You would count the plaques and then be able to extrapolate how many viruses you had in that sample. It's kind of the opposite with bacteria. We might count the colonies. Here we're counting where the bacteria have died. It's also very common to grow viruses in eggs. Eggs are really just great big cells. And depending on what virus you are dealing with, you might want to inject that egg in. There's lots of different spots that you can inject that egg. We grow some of our vaccines, like the influenza vaccine, in eggs. So the influenza virus, we grow up in eggs, and then we treat them to inactivate them in order to make the vaccine. This is pretty time consuming. I don't know of any of the coronavirus vaccines that are grown up in eggs. We actually have to decide on the formulation of the influenza vaccine in February for the following flu season. So it's quite a laborious process. If we could tweak that process so it would take less time, we could better formulate our flu vaccines and ones that were matching better the circulating strains. Okay, and another way that we grow viruses is in cell culture. Okay, we used to refer to this as tissue culture. Really, tissues are supposed to be several different kinds of cells put together, so it's better named cell culture now. You basically put the media, into
Hey, Professor, I don't know if you can hear me, but everyone on Zoom can't hear you.
all right. Well, we will continue. And yeah, okay. I'll have to figure out what to do. Thanks for letting me know, though. Okay, so that's pretty much kind of wrapping up thinking about viruses. If you think about, like I said, this is something that people argue about, are viruses considered alive or not? Now that we know some of the characteristics of them, thinking about the things that we would have to satisfy to consider an organism being alive. Okay, an organism that's considered alive would grow, it would reproduce, it would respond to some kind of stimuli. Yeah, oh, right, okay. I forgot that I have to do that. Okay, I have to invite myself again, sorry. You know what? This is probably a good time to stop. We only got 5 more minutes. Would we agree with that? Okay. All right. Thanks for your patience. We'll start into chapter 9 next week.